Hey everybody, this week we are talking about water. Normally throughout this lecture I have videos that I show to demonstrate a few different things. I'll try to play little snippets of those videos in here, but the longer ones I'll just link for you to watch separately so it's not super distracting. We're looking at water in plants, determining where plants live and how they function since the beginning of time. This is one that I'm going to link for you to watch. It's a little bit longer. David Attenborough, favorite guy, walking through this process of how water moves through the plants. And that's actually what we're going to walk through in the lecture today. So if you want to stop and watch that first, I would recommend it because it'll give you this visual map of the process, which is pretty fascinating. Plant tissue is made up of 90% water. You might often hear the statistic that humans are made up of 70% water. Plants and fungi too, 90%, so almost entirely water. Maintaining that volume of water in their bodies helps them keep their form. So it is incredibly important to the overall structure of a plant as well as the function of a plant. The stature of non-woody plant tissue is achieved by overinflating the cells with water. So we can think about how plants do this. And normally the lab that I do for botany this week that corresponds with this lecture, we do all of this like stuff with solutes and shrinking and growing the vacuoles of plant cells. I took that out of this week's lab because I wanted to focus on the experimental design that you'll be working on. And it felt too disjointed to have that component and then this other experimental component. So if you want to experiment and see what we're talking about, you can take an Lodia leaf off of your Lodia, put it on your microscope slide, mount it with regular water, look at it under the microscope, soak up that water, add in some salt water, watch it under the microscope, watch as the central vacuole shrinks in. That pulls the plasma membrane in, and the whole internal contents of the cell collapse, but the cell wall stays the same. So you can watch this sort of change in the cell's structure based on this change in salinity. And that is going to have a lot to do with how plants function and where they can grow. So the way that plants overinflate their cells, I'll get to this part, is by putting a bunch of solutes inside the central vacuole. So the central vacuole inflates and kind of pushes the rest of the cell contents outward, making that cell turgid, and that gives it structural support because it's inflated. To do that, plants need to be able to suck in water from the exterior environment and overcome the sort of pressure that would want to push that water out by wanting that water to come in chemically, right? So they have to lure it in by filling the central vacuole with solutes, like dissolved sugars. So many compounds move through plant cells via diffusion. This is the passive movement of molecules to areas of lower concentration. So this is a concept you should be familiar with from cell bio, that molecules want to move from areas of high concentration to low concentration. And when we learned about photosynthesis last week, you kind of addressed that concept with that flow of protons. That the protons were being packed into an area of already higher concentration, didn't want to be there, and would use the concentration gradient to produce energy and flow down that concentration gradient. So molecules just naturally, chemically want to do that. When water diffuses across a membrane, it's called osmosis. So this passive diffusion of water from areas of high water concentration to areas of low water concentration. And it's going to be relative to the amount of other stuff in there, the solutes. And as it goes through a membrane, that's called osmosis. So what we would normally do in lab would be to demonstrate this water movement, how and why water moves, and to really visualize that by changing the solute concentration of the solution that something is in. So imagine an egg as a big cell, right? Because that's what it is. It has this nice cell membrane. You can think of the shell as the cell wall. And then I like to think of the yolk as the nucleus because it's just this large central structure that we can see. So in this experiment here, there are eggs that have had their shells dissolved off by vinegar. You just put an egg in some vinegar, and after a couple days, the vinegar interacts with because the vinegar is acidic and the shell is basic, so it just reacts with each other to produce water, CO2, and some salt. And so it dissolves the shell off of the egg, and you're left with just that thin membrane that's on the inside of the shell. That's very much like the plasma membrane. So now you have two giant cells you can experiment with. And in this experiment, they are going to take those eggs, weigh them, or get their mass, and then they're going to put them in different solutions. So you can put them in a hypertonic solution, and hyper, more, a lot. Tonic, it has to do with dissolved stuff, contents. So a hypertonic solution would have a lot of dissolved stuff in it. So that dissolved stuff could be sugar, like a corn syrup would be a hypertonic solution relative to water. It could also be salts. So if it has a high salinity, then that is going to be a lot of dissolved stuff in there. Again, hypertonic relative to regular water, or the contents of that egg. If we put it in a hypotonic solution, that would have a low concentration of solutes. And so that could be something like deionized water, where it would have a low concentration of solutes. I'm trying to think of the other term. There's like a, I don't know, maybe deionized water is just the term I'm trying to think of, and I thought of it. So I'm going to play a little bit of this video and sort of jump around through it.
What happens to a cell when it's placed in a hypotonic solution versus a hypertonic solution?
Okay, so these are the eggs when they're in the vinegar.
Calcium carbonate will break
It's
down
going to dissolve the shells.
Hypotonic solution.
There's the shell dissolving, forming lots of CO2 bubbles.
Within 3 days, you should have a semi-permeable membrane.
But here comes the interesting part. So deionized water and a hypertonic corn syrup. See how this egg is shrinking. The water is leaving this egg and going to a place of relatively lower concentration. So the water in the egg has a relatively high concentration of water relative to the solutes in the egg. Outside of there, there's tons of dissolved solutes in that solution. It's very thick, full of sugar molecules. So the water is going to leave the egg and go to a place of lower concentration relative to solutes. So it's trying to even out the amount of solutes to water within the egg and within the external solution. Within the hypotonic glass over here, there is relatively low solutes in the exterior and relatively high solutes inside the egg, so water is rushing from the outside into the egg, causing it to swell up. So this egg is getting a little bit bigger, and this egg is actually getting a lot smaller.
Egg A now has a mass of 97 grams. Egg A increased slightly to 103% of its original weight. We can conclude when a semi-permeable cell is in a hypotonic solution, water will enter the cell. Egg B now has a mass of 59 grams. Egg B decreased 57% of its original weight.
So that's a huge change. This is also really fun to do, especially if you have kids or just curious people around. It's a cheap, easy experiment. Easter is also soon. If anybody wants to do stuff with eggs, I highly recommend doing this. I probably just should have put it in the lab. But you can put dyes into the solution, and if the dyes are small, they will make it into the egg. If the dyes are large, they won't. So it's a fun kind of way to see this process of diffusion in a more colorful way. So let's see, they're then going to take these two eggs and swap the solutions that they're in. They're going to put their hypertonic egg into a hypotonic solution and their hypotonic egg into a hypertonic solution, and then see the change.
B is going in the hypotonic solution, which is still on the left.
Oh, look at him go! So this is just a fun way to visualize osmosis and how it works. Okay, so plant cells can change the composition of the central vacuole to promote the movement of water into the vacuole, making the plant cell turgid by concentrating solutes within the central vacuole, drawing water from the outside and expanding like that egg that was in the hypotonic solution. That's what plants are doing. That's why when plants are in a like salty soil, or a soil with a bunch of solutes in it, it doesn't have to be salt, it'll draw the water out of the plant. Plants can't really survive in a saline environment or a high solute environment unless they have specially adapted for that. So this is part of what determines where plants can live, and plants that are trying to exist in a saltier environment, like plants that have moved back into the ocean, have to have special capacity to be able to store more solutes and kind of compete with that exterior saline environment. Okay, so let's look at these turgid plant cells. This central vacuole full of water is pushing all of the cellular contents out toward the wall. The wall is this rigid structure that holds it all in and keeps that cell from bursting. If this were to happen in animal cells, the cells would burst. So our cells need to be in an isotonic solution. They have to be in the relatively same concentration of solutes as the solution they're in. So our blood cells are in our blood plasma, and they are isotonic. That keeps them in their normal condition. If they were to be in a solution with relatively low solutes in it, like if you got an IV that didn't have the correct amount of solutes and they were too low, the water from that solution would rush into your cells and burst them. If your cells were in a concentration, or if you got an IV with a concentration that was too high, then the water would rush out of your cells and cause your blood cells to shrivel, called crenation. Here is a cool example of just the central vacuole and how big it is within a plant cell. So these are the little kind of hairs that are around the stamens of a plant called Tratiscantia. And so it's got these purple stamens, and you can see here they're made up of tiny little, well actually they're quite large, so you can see the cells. They're just these huge cells that are kind of stacked on top of each other like little beads on a chain. And so if you look at those under the microscope, you can see them here. Each cell has this huge purple, it looks like the whole cell is purple. But remember that anthocyanins are that purple pigment, and they're stored inside the central vacuole. So this shows you the sort of delineation of the central vacuole and how much of the cell it ends up taking up. So you can see the nucleus in here that's connected to the rest of the cytoplasm by these little cytoplasmic strands. But the central vacuole is filling up almost that entire cell. So plant cells are highly occupied by the central vacuole that gives them their structure. If that central vacuole were to shrink because it wasn't able to keep a high water concentration, the whole structure of those little hairs would just flop down, right? That's when plants wilt, is when they're not in that turgid condition because they're lacking water. This is what it looks like in your Lodia. The central vacuole will start to pull away from the plant cell wall. So here's the boundary of the plasma membrane. Sorry, the plasma membrane. As the central vacuole shrinks, it's the thing that keeps the plasma membrane pushed out. So it starts to pull away from the cell wall. So here's our cell wall, here's our plasma membrane, and then our central vacuole is just inside of there. I think this is where the nucleus is, right here. And you can see what happens. You might be familiar with this if you're as bad of a houseplant keeper as I am, but a plant that is in a turgid condition that has plenty of water, cells are overinflated, will look like a normal plant. A plant that is low on water, doesn't have the overinflated central vacuole, will be wilted and sad looking. And we think of these plants as dead or dying because they've lost their internal structure. Okay, so I'll stop here and continue with roots in the next video.